We're gonna continue with the most interesting topic of all, of course, which is output tables. And because this is, this is the one thing one actually has to know or understand to work with MaxQuant and maybe also Perseus if you want. So I'll keep this relatively simple and basic, but I think I will tell more or less all interested people what you need to know. So what are output tables? And this should be the typical workflow of a project. So basically, what we do is we develop these two softwares. We develop MaxQuant and Perseus. And this is kind of the workflow you can do with these two softwares. So you get the raw data from your spectrometer, and then you do crazy analysis with MaxQuant. Basically, MaxQuant does the crazy analysis and you press a button. And then in the end, you get the output tables. And these output tables are the link between like MaxQuant and what it does, all the magic there, and then your analysis, which you can, of course, do with Perseus, and we like that. But you can, of course, also do with whatever self-written Python, MATLAB, R, whatever script you want. So the output tables are the one thing that connects our two softwares. And mainly they serve two purposes, or maybe you can approach them from two perspectives. So on one hand, from a scientific perspective, so if you're actually a scientist, so these are the stuff, the data that you analyze. So this is the stuff you need for your statistical analysis. But on the other hand, if you run a core facility, for example, or you want to optimize your method, you want to find out if your spectrometer works nicely and so on, you also can use these output tables for your, basically for benchmarking your method, your machine, for quality control, method optimization, and so on. So these are the two main purposes, and depending on which side you come from or which approach you have, you might make use of different output tables. Okay, so let's start. First of all, maybe most importantly, is where actually are these output tables? So this would be your project name, and then there's a folder called combined, and then another folder called txt, which stands for text file, because all these output tables are tab separated files. So you can open them basically with any text editor you want, but also with Excel, for example. So, or whatever other stuff you want, but they are human readable, all of them. And what is already highlighted here, but I'm going to highlight it even bigger so everyone can see it, is this PDF here, which is called tables.pdf, which is actually an explanation of all these tables that are generated by MaxQuant, because they have a lot of columns and it's not always easy to understand, or you don't always memorize exactly what this column means or the other column means. So here you can find explanations for everything. And it looks like this. It's a very long PDF document. So here the bar is very small, so because we have a lot of columns. So this is where you can find your output tables. And then you can read them, as I said, with Excel or with whatever you want. This is how it looks like. Okay, you can, of course, read them in Perseus, and we're going to start today with the Perseus tutorial. So I'm not going to cover that, how to read an output table into Perseus, but we're going to do that multiple times today and tomorrow. But this is just a first glimpse of how that might look like. But you can also visualize all your data that is then saved in output tables in MaxQuant in this visualization tab, which is here basically, and then you can have all kinds of visualizations. So if you have this active here, then you're going to see four little tabs here on the right side, which I'm going to make a bit larger soon. And these correspond to the four logical kind of groups that we grouped our output tables in. So what I'm going to do in this talk is basically go through these groups step by step and talk about the different tables that there are and might be interesting. So you can see them here. And I'm going to enlarge on that. So we have these four different groups, levels, whatever you might call them, clusters. And I'm going to structure my talk based on that. So what do you have? First, you have the scan level. So what does scan level mean? So basically, you can get information about your different MS scans, MS1, MS2, MS3, if you have MS3. And this is mostly interesting for quality control. So if you want to find out, did everything work well? What could I optimize and so on? So this might be mostly interesting for people from core facilities, methods developers, and so on. Then we have the identifications tab, which includes most, or like is the largest group of output tables. And this is the group that is mostly interesting for the actual scientific people. So this is mostly the data that you will probably analyze statistically and that you are interested in. So what is there? There's information about the protein groups that were identified, and then the peptides that were identified and gave rise to these protein groups, PTMs and so on, MSMS level, but also evidence. So what is evidence? Evidence basically is giving you information about all the peaks that were leading to a peptide identification. But I'm going to talk a lot more about evidence very soon. So you get everything that resulted or is related to an identification in the end. Then you have the features level. What is features? So basically not everything we see in our data will in the end lead to an identification. Maybe the Andromeda score was not high enough or whatever. So here in this feature, two feature tables, so all feature where you can see this, basically there you see all the features. So all the stuff in the data that was identified as some pattern, but did not give rise to a peptide or protein identification. And then lastly, we have this metadata tab where you can basically see some overview parameters for quick quality control and so on. So I'm going to go through that now quickly, or not quickly, slowly. And I'm going to start with the identifications because I guess that is most interesting for people here. So maybe we start with the protein groups.txt document, which gives you information about the identified protein groups. So which protein groups did I identify? Which properties do they have? Which proteins are actually in this group? Which peptides are involved in that? And so on. So you get a lot of information. So exactly. So properties of the protein, like which amino acids do these proteins have and so on. Peptide counts. How many peptides are involved in this protein group? So how many peptides gave rise to this identification? How was it identified? Matching between runs or MSMS. And then also, which is quite nice to know, is like how much of my protein was actually covered by peptides. So did I see only 5% of my protein or maybe 50%? So this is then the sequence coverage. Exactly, you can compare peptide counts between different runs of your experiments and of course get all the scores. So this for the identification, but then if you want also quantitative data, you get all the intensity data, LFQ data, and so on. So this is how it looks like, and I'll try to change now to show something like this. Exactly, so I made it a bit larger. I hope everyone can see it. So of course, it's not always yellow. I just made it yellow because I think this is the most interesting stuff. So what do we have? I mean, now we have the protein groups table. So the first information is basically which proteins are in my protein group. So it just gives you the UniProt IDs of the proteins that are involved in this group. And they are sorted by basically score. So how many peptides did I find per them? So the more peptides I found per protein, the more left it is, and so on. So these counts are then here. So how many peptides per protein did I find? This is for all peptides, so both unique peptides and razor peptides. And the order, if there are multiple values in here, is always the same order as for the protein IDs. So you can actually match them together because the order is always the same. Exactly. So then there's a lot of self-explanatory stuff, like the gene name of the protein proteins in the protein group, or just the first protein, which is most likely to be it, and so on. Then the number of proteins in your group, the number of peptides, then divided into unique peptides and razor peptides, and so on. Then what we can see here is the same again, so peptide count. But now we can see that there were 3 different cell lines used in this experiment. So we have this here, HCC 1143, 2218, and then, yes, another one here. And per cell line, we had 3 repetitions, so _1, _2, and _3. So that's what that means basically. Exactly. So how many peptides in this run of our experiment for this cell line did I find? That's the information you have here. And then again for, so this is combined, razor and unique, and so on. Okay. Then, as I said, we might be interested in the sequence coverage. So this is now for the only the very first protein in your protein group, because it's just one value. So when it's just one value, it's only the first one, which is most likely to be the most interesting one. So you can see that here. And then again, the coverage for only unique and unique plus razor. Self-explanatory stuff like molecular weight, sequence length, and so on. And again, like here, just for the first one, and then for all of them that are in this protein group. Then some self-explanatory stuff. You can, of course, get the Q value, the score, and so on. So Q value, not this Q number that we had this confusion with Pili. So Q value is the corrected P value after you did your FDR control. So just don't get confused. And then here we can see how was it identified in our different runs. So we can here see again, okay, for this cell type in our second run, how did we identify it? And it should be either by MSMS or by matching between runs. That's basically the 2 options we have here, or of course not identified at all. So, and then we can see here, because we have a ratio between heavy and light, that it's a Silac experiment. So that's like, if you have a Silac experiment, then this column will appear basically. And then again, like normalized and so on. So a bit more about that. Exactly. So then some, I mean, just split up again for all the different runs and so on, but it's all very self-explanatory. And then in the end, you have the intensity. So this is the total intensity, basically summed up over everything. And then here you have basically, again, the light channel, heavy channel, and this is the sum of them. Okay, for all your different runs and cell lines again. And then in the end, what we have is one ID per group. So how we do matching between different tables in Max Quant or Perseus is basically every protein group and every peptide and so on gets a unique identifier for our experiment. So it's basically just numbered. So here this is identifier 0, 1, 2, and so on. And then every peptide has the same. So what we do here is just we match to which row in other tables we should match our protein group here, for example, to which rows in the peptide table, basically. So we had seen before that there were 4 peptides involved in this protein group. So we have now here 4 peptide IDs that we might look at in the peptide table if we want to. So that's basically just how you can reference from one table to the other one. Okay, and then we have this for all kinds of other tables. And this is always for all tables at the very end of the table. So now the table is over. So the structure is always the same. So this ID and then the reference to the other tables, they are at the end. And at the very beginning, there's always the stuff that like identifies what you're looking at. So here, for example, the protein group, it makes sense to have first the proteins that are involved in this protein group. And now I have it already open. I'm going to just quickly show also some other tables. So I talked about the peptides table already a bit. So we had here 4 peptides involved in this identification. So we can look at the peptides table. and then go to the far left. And here, I mean, on a peptide level, it doesn't make sense to have some protein IDs or something, but here, what actually makes or identifies this peptide is basically its sequence. So that's the stuff that constitutes a peptide. So this is the first column. And then you get information about cleavage sites. So the N-terminal can also be empty, like here. Why is that? Because basically, if this is the very N-terminus of your protein, then it's not possible to have a cleavage site before that. So that's why sometimes these columns here contain empty, like underscored value. So that just means that your peptide is at the very N-terminus of the protein. And yeah, so you get the N-terminal cleavage site, C-terminal cleavage site just meaning that here in the middle there was cut, and this is the region around where was cut in the protein. So you get the amino acid before cleavage basically, and obviously this, if you digest with trypsin, should be these two letters. So if not, then you have some like you could ask what's going on there. And you can also check by your sequence of your peptide if there were any miscleavages. So if there was some possible cleavage site missed. Then you get a lot of information about the amino acid sequence and so on. So just the properties of this peptide. Here you see the last amino acid of your peptide, which again should be the same as the, so it should only be triptych if just like in this column, because I mean, it was cut after your peptide ended. So it makes sense that it should be this. Exactly, then some just amino acids counts and so on, and some simple properties like length, miscleavages, exactly. So we already summed them up for you. Then the mass and the protein that is like the first in the protein group. So if there are multiple ones that they match to, then you get the leading razor protein, the start position on this protein, end position of this peptide on the protein. So if you want to see, okay, where exactly am I in my protein? Then some redundant information from the protein group table, so gene names and so on, if you want that. Then charge states, so it can be multiple ones because maybe we had different charge states of this peptide in our data and then was collapsed, as Pili told on Monday. So it might be just one charge state, but maybe it's also multiple ones, the PEP scores and so on. You see again the identification. So it's very similar from the like construction and architecture of this table to the protein groups table. And then intensity values and so on. And then, as I said, again, in the end, you get these IDs. So these are now the IDs for the peptides. So every peptide has an ID, just like the protein groups. And then you get one protein group ID, which this peptide belongs to. So this references again back to the protein group table. And of course, in this case, it's only one because every peptide, like in most cases, it's only one because typically one peptide is matching to one protein group. And then some references to other tables if necessary or if interesting. And then lastly, because I guess this is also something that people are using a lot, is basically the table that you get when you are interested in in modifications, basically. So here you get the proteins where these modifications might occur, and then the position in these proteins where this modification did occur. I mean, that's the information you want to have actually. And then again, the most likely protein, so which of them is more likely to be, the name, gene name, and so on, as we just had. And then what is really interesting is this localization probability. So how likely is it that this localization of the modification in the protein is actually correct, that we just told you? And here you might define your own cutoff if you want. So I mean, the probability of 1 is extremely good, but there are also some values that are really low. So you might consider, okay, which ones do I actually include in my data analysis and which ones do I not include? Okay, then again, scores for each localization and so on. But what is most interesting then in the end is this. So basically you get the sequence. And for every possible site where we think there might be one of these modifications, you get the probability score. So here, for example, we are Max Cohen is very unsure where exactly this modification did take place. Whilst in this example, for example, I mean, there's only one possible modification, so Max Point is very sure where it can be. And then in the end, you get these IDs and so on, as we already had before. So these are some examples of our tables, and I will go back to my PowerPoint. Okay, so that's what it looks like. I tried to show it to you just now. We had the PTM sites table, so you can get all kinds of information there, which is more or less in the same structure as in the protein TXT table. So these two tables are probably the ones that you are most interested in if you are an actual scientist and you want to analyze your data. So typically these two are the things that you are interested in. So if we want to understand the other stuff, good. We have to look again at how our mass spectrometry-based proteomics works. So what do we have here? What do I mean by evidence and so on? So, I mean, we heard it now a lot, is that we have these proteins group, on proteins are digested. So actually we have peptides that we have in our data, and actually we don't have peptides, but we just have these funny three-dimensional plots of mass over charge ratio to retention time. And then there are these funny little peaks that we have here. I mean, that maybe you remember from Monday was a picture that Pili showed. So what we want to do is basically we first want to identify each peak here, and then we want to do a second MS on this, and then hopefully we will match the spectrum to one of the peptides in our database. And we don't want to do this just once per peak, but it would be nice if we would do this multiple times per peak, because then we would have a lot of evidence that actually, yes, true, this peptide is in our data. So evidence means all the peaks that in the end contributed in the identification of our peptides and then our protein groups. So this is what evidence actually means. So each peak is a feature, and if this feature contributed to identification via second MS to one of our peptides, then we call this evidence. If it did not contribute to identification, then we just call it a feature and not evidence, because it did not contribute to anything. So we saw the peptides tables. I'm just going to skip this. So, but what can we do with the data we saw? So maybe we just want to plot, okay, does our data make sense? So for example, did we cover different lengths? Did we cover typical lengths? Are there gaps that there? So maybe we have some systematic problems that some lengths are not allowed, whatever. Then we can plot the miscleavage rate. So we of course want this to be very low, that we missed some cleavages. And we want this to be high, so we can use this to test our digestion, basically how good did that work. Then we can look at the cleavage window. So did it actually cut at the correct positions? Does it make sense? Does it match to our protease? And then we can look at the coverage. So basically we have for every peptide, I showed you the start position and this end position. So we can plot something like this. So is our stuff covered, or is it just in some regions of the protein where we have peptides? So we can plot something like this, or we can plot the intensity for each of one of our peptides against the start position. So for example, here we can see that somehow the second half of this protein that we have there, that the intensity is much higher than for the first half. So maybe the second half is actually more abundant in our data set, which might be interesting to know, for example. So this you can do with the peptides table, which I just showed you, and we saw that. So there is like also information how many features, so peaks, contributed to this identification. So these are the number of features that contributed, and I just said that evidence is basically each such peak. So you might remember more or less the slide. I mean, a bit different, but the same pictures from Pili's talk. So basically we have this mass to charge over retention time, three-dimensional plot where the z-axis is then intensity, and we have this three-dimensional peaks. We have this isotope pattern here. and distribution over retention time. And then we identify the peaks, we align them and so on. And in the end, we get one of these nice three-dimensional peaks. And we call one of these three-dimensional peaks that we have there a feature. And again, if this feature contributes to identification, then we call this evidence. So what we get in the evidence file is basically all kinds of information about our peaks. So we get information like, okay, how many did you identify and so on, of course, but in which charge state were they? Are they modified or not? How did we identify them, by a second MS or by a match between runs? And then what's the score? What's the retention time? What's the mass error? How good did recalibration work and so on? So what can we do with this? So it looks like this. So what can we do with this? So for example, we can plot the mass error against retention time for different runs of our experiment. So for example, in the experiment I had in my example table, we had 3 cell lines with 3 runs each. So then we might be interested in, okay, are they comparable between these runs? So what we can do is that basically we plot something like this, retention time against mass error. Of course, you should do this before max quant recalibrated, because otherwise it will all look nice. But you can see, okay, this is nice. It's all distributed more or less normally, and it makes sense. Wells here is kind of wobbly and weird. So what might cause this? For example, maybe someone walked into the room with your mass spec and opened the window. Maybe you have different experimenters doing this and so on. So by this, you can actually find out if your experiments are comparable between each other. Then exactly, you can do the same with the retention time. So you can calculate correlations between retention times between different runs. You can plot the retention length and see, okay, are the distributions for different columns the same or not? And if not, you should maybe look into your data again and your method again. So all this is in the evidence table. And then again, not again, but you can also look at, okay, if I identified something, how many sequences, like how often did I have to do a second MS? So it should be optimally either 0 or 1. So 0 is basically identifying by matching between runs. So you didn't do a second MS, but you identified it because in a different run you did it. Or 1 because you have done second MS. And if you have more than 1, then something went wrong. So basically then your fractionation window didn't align correctly with the peak, so you had to do it again. Exactly. So that was the identifications group of tables, which I think is most interesting for most researchers. And now we go to the scans group or level of tables, which is more interesting for quality control and method control. Okay, so what's that? So you get all the information about your different spectra, basically, and your scans that you have. But this is not always there. So you actually have to switch it on in MaxQuant if you want these tables. So you go to global parameters, and there you tick this box, calculate peak properties. So only then you get this, and you can switch it off if you don't want it, basically. So what can you do? So for example, you can look at, okay, for my precursor selection, which intensity, if I choose which intensity of my precursor, when did it actually still result in an identification at the second MS level? So you can do this relative fraction. So you take the peak or the apex basically of that peaks. So you set this to 1, and then you look down and say, okay, at which ratio relative to this peak, if I did second MS, so over your retention time, did still lead to identification. And so you can say, okay, if this is very low or very high, then should I do this? So if this value is very low, but you set your experiment, your method that it still should do second MS if intensity is very low, then maybe it's not worth it. You're just wasting time and energy basically on this. So this tells you something how to set the parameters of your spectrometer. And you can do this in retention time. You can, of course, also do this in mass over charge. So that's basically the same. So you look at the highest peak, and the question is, okay, if you do data dependent proteomics, so which peaks should I select? So relative to the highest one, should I select only like very stringent cutoff, so only if it's like 50% of this highest peak, or maybe very low. So here you can get all kinds of information at which relative fraction it still led to identification. And this is in this second MS scan table, basically. So if it's a high value, then probably you shouldn't do second MS for very low peaks, for example. Exactly. And then you also get information about your isolation window. So how many, like the fraction of intensity that actually was part of your target ion. So and how much was just a contaminating ion. So also that information is in the second MS scan level. So that you might optimize how wide you should choose your isolation window. And this might be interesting for people doing TMT labeling. Okay. Exactly. So what can you do? You can also look at, okay, at my first MS level, I have this intensity, and at my second MS level, I have these multiple intensities. So if I add all these up, these should be more or less the same as the intensity before, because it's just it's the same but fractionated, cut into pieces. So if you sum all these peaks up, they should be more or less the same as this. And you can just plot that against each other. So it should not look like this. So then it's like too high, and it should not look like this, then it's too low. So it should optimally be on this line. And if not, then maybe you should reconsider your parameters. You might also get information or want to look at information about the cycle time. So what's the cycle time? We have here one of these peaks in the retention time. So we had these 3-dimensional peaks, and this is now basically in a projection in 2 dimensions where we just look at the retention time. And if what we do basically, if we do data dependent search, is that basically your window goes from peak to peak depending on your settings. So of course it needs some time to go to different peaks. So the question is, when does it get back to the same peak, to basically be at the same peak in the master charge axis, but be further down in the retention time axis? And what we want in the best case would be that we have multiple samplings from the same peak, because only then we can do statistics. So we want multiple samples from the same 3-dimensional peak to actually be relatively sure about the data that we generated and about our identification. So what we want is a relatively short cycle time, and cycle time meaning the time between measurements of the same peak in retention time. And we can plot this. So this is, for example, the time of your experiment. So basically how long you ran your experiment against cycle time. And if it looks like this, it's very bad. So at the beginning, the first third maybe of the experiment, cycle time is relatively low, but also so this is the MSMS count. So basically how many second MS scans did you do? So both is short. What does this mean? It means that there are very few peptides there, because you have very few second MS scans, and this then makes your cycle time very short. So in the beginning, there's always no peptides, and then both jump extremely high all in a sudden. So what that means is that in the beginning, you have almost nothing, and then everything in one bunch. So you can't do, I mean, your machine can't work that fast. So your cycle time goes up, and that means that you get less data points per peak, basically. So you might play around with your gradient to optimize this, to like pull this a bit further down and this at the beginning a bit further up. Exactly. So if it looks like this, not optimal. Okay. So let's go to the features level. And features now, again, is the same as evidence. So every peak in this multidimensional space is one feature, and only if it contributed to peptide identification, we call it evidence. If not, we call it a feature. So we had this again, this is a feature in the end, after all this process, one of these peaks. So what might you now be interested in for quality control, for example, is, okay, what's the difference between features that we could identify and features that we could not use for identification of our peptides? So again, it has the same kind of information like charge, mass over charge, intensity, and so on. How is this peak shaped and so on? This is more or less the same as in the evidence table. And then you can do something like this. So gray is all the features that did not contribute to any peptide identification. And these are the ones, so red are the ones that were then targeted for a second MS, and green is the one where actually something was identified. And of course, you want to be this more or less the same. So you can check different parameters, like different settings of your machine and so on, and then check, okay, how did this affect these kind of plots, and find out what is optimal, for example. Exactly. And in the end, we have this metadata output that just gives you a summary of your data, and also it might be interesting for just a very quick quality control and also comparison between different machines or maybe different experimentators if you want to do that. So it gives you all the high level information like number of identified peptides, number of identified detected isotope patterns and so on. So more like summary statistics about the quality of your experiment. And so this is basically in this table called summary.txt. I mean, that's exactly what it is. So it's a quick overview of what you did. And then what, for example, you might do in, for example, a core facility is you might define yourself in this table some kind of metric. So maybe you just want to sum up different scores or whatever. So you define your own metric, how to measure quality in this, and then you can compare them between different experiments, between different instruments, between different experimentators, between different days, and so on to basically stay at the same quality level over time, more or less. So that might be a quick and easy quality control. So now this was a lot of tables. You don't always have to generate all of them. So you can go to this global parameters and there's a tables tag basically. And here you can select which tables you want. So this is, I think, an old screenshot. So now there are more options. So you can unselect more tables if you want, if you don't want all this bunch, but fewer ones. And with that, I'm done with my talk. So we have seen that we have 4 different groups, identifications, which includes most of the, let's say, interesting scientific data, then a lot of stuff that you can use for quality control, and an easy method to summarize and compare experiments. And that was it. Thank you.
Thank you very much. Is it possible anywhere in Perseus or in MaxQuant visualization somewhere to get some of these quality control like plots?
So not automated in exactly the same way. So of course you can do all kinds of plots. You can do histograms and scatter plots and whatnot all in Perseus, but you still have to click yourself there. So it's not automated like do me this and that quality control pipeline or something.
Okay, thank you.